Assalamu alaikum wa rahmatullahi ta'ala wa barakatuhu. Welcome back again to our biology class, genetics continuation of the chapter. And today, as you can see on the board, we want to navigate through the first law of heredity formulated by the Geiger Mendel. Now class, as you can see listed the law, then the way whatever he used, and now and the successful the things that made his law to be successful and one that is currently used in heredity. Now class, as you can see, we say the characteristics of an organism are determined by internal factors. The characteristics of an organism are determined by internal factors which occur in pairs. Only one of a pair of such factors can be represented in a single gamete. And class, as we have been all along doing, we are discussing about inheritance, variation, chromosomes, DNA, genes, all those things. But today we want us to look at the law of this heredity. And we said the hereditary materials are contained in the nucleus, particularly the long molecule that is the DNA. And the DNA is found in the chromosomes, and the DNA contains subchapters called the genes. Now today I want us to look at the law of heredity. That is the first law of heredity formulated by the Geiger Mendel. As you can see, the characteristics of an organism are determined by internal factors which occur in pairs. Class, internal factors basically means that these characteristics are found inside, which occur in pairs. Only one of a pair of such factors, and we said that only one of a pair of such factors can be represented in a single gamete. For instance, class, let's say we have, let's say, a characteristic for tallness, for homozygous tallness, like TT. Now class, that is a homozygous tallness, but again we are going to see this is now the genotype, but before fusion, these two are going to segregate into different gametes, and therefore we are going to see that this one is going to be T like that, while this one is going to be T like that. Now let's go back again to our definition again. We said the characteristics of an organism are determined by internal factors. These being internal factors, they are found in the chromosomes, which is found in the nucleus, and the nucleus is found inside the cells, and the cells are found, they are internal. They are very, very good. Now let's move to the next part, which occur in pairs, and we can see that this genotype for tallness, which is homozygous tallness, is a pair. And again, this pair, for it to undergo the process of fertilization or rather fusion, it has to segregate into different gametes. And therefore we say one of such a pair can only be represented in a single gamete. Hope that is very clear. What I'm trying to say is the characteristics of an organism are determined by internal factors. Why? This because the factors which determine the characteristics of organisms are found in the nucleus of a cell. And the nucleus of a cell is found within the organism, and therefore that is being internal. Again, they occur in pairs, and we said that characteristics, this genotype, they occur in pairs. This is homozygous tallness, for instance. And therefore again, during fusion or fertilization, this genotype is going to segregate into gametes, and therefore only one of a pair of such factors can be represented in a single gamete. That is done. Now, Geiger Mendel is the father of inheritance and the law of heredity. Now, this man, he employed some mechanisms and brought these crosses. Now, what he did this man was, number one, he self-pollinated dwarf plants. Dwarf plants basically means short plants. After self-pollinating, the seeds were formed. Then he took those seeds. Then he planted those seeds. After planting those seeds, he did observe what was their height. Now he therefore observed that all these seeds grew up into dwarf plants. That is what he made as an observation. And therefore we said that these dwarf plants are pure breeds. And therefore class, for us to say a plant is dwarf, dwarfism can either be like this, TT, that is recessive. That means homozygous recessive. And therefore class, this one is the only time that dwarfism can phenotypically expressed. But other times, in a case like TT, this one phenotypically is tall, but genotypically is containing a gene for tallness, as well it also containing a gene for shortness. Despite this gene for dwarfism existing in the genotype, does not express itself phenotypically, because we say this one is a dominant gene over these. And therefore, this one expresses itself phenotypically, but the gene can be transmitted to the offspring during fertilization. Now this man is self-pollinated some dwarf plants, and therefore class, when we say a dwarf plant, we totally mean that condition of the recessive, the homozygous recessive, like that. And therefore class, if you cross this homozygous recessive with another homozygous recessive, crossing dwarf plant basically means you're crossing a homozygous recessive with another homozygous recessive, and of a surety you're going to get all the plants to be dwarfism. Very good. Now that's your case number one. After self-pollinating them, he took up their seeds, planted them, repeated the experiment, and found out that all the plants that grew up were all dwarfs. Right? That is number one. Now again, in this next approach, what he did is he self-pollinated tall plants. After self-pollinating tall plants class, then what he did was, again he took the seeds and planted them. Now this one, they did show some variations. We were having some plants which grew up tall. We are also having others which grew up to be dwarfism. Now class, this man, he found out that this one was still, he didn't got his accuracy that he was looking for. Again, he took the pure tall plants, then he selfed them, then until he got pure breeds of tall plant, And when he got the pure breeds of tall plants class, now that means, why are we having possibilities here? Class, why? This is the reason why. Because when you have a condition like this, TT, a condition like TT, this one indicates, this one can result in a plant being tall, right? As well, this one can result in a plant being tall as well. But the problem again is, this one was tall and this one was tall. But which one was it crossed? And therefore, that is why we are having possibilities that when tall plants are selved and their seeds planted, we can have plants growing tall height, others growing short height. Because in the genotype of tallness, we can still have a gene for shortness, which is transferred to the offspring, and as a result, the offspring becoming growing to shorter height. That is one. Now, that is case number two. Now, number three, what he did was, and he repeated the process until he got pure tall plants. Now, what he did was, he cross-pollinated pure-bred tall plants with pure-bred dwarf plants. Now, what he got again, he got all plants to be tall. From there again, pure-bred tall plants with pure-bred dwarf plants. Now class, let's look how this thing was possible. From number one. Let's start with the first one. Now we say he crossed self-pollinated dwarf plant, resulted, now we say like this, we have parental phenotype. Which is dwarf, still have dwarf. Parental genotype. Parental genotype will be TT, and you must show that cross. Again class, what we are calling, each of such pair is contained in a single gamete. Now, let's cross this one. It will be like that. Now, class, these are the gametes again. These ones are the gametes. This is the process of fission or other fertilization. Now, let's look at the offsprings. Offsprings. Now, in the offsprings, we have all offsprings will be dwarf. Again, recessive homozygous for dwarfism. TT, 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 TT. Now, as you can see, all offsprings are dwarf. I think. Now, if you again take these seeds, then you plant it, you're still going to get the same. Why? Because there is only the gene, all these plants are homozygous recessive. And therefore, now, phenotype, F1 generation, phenotype, dwarf. Well, the genotype is TT, 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 TT all. Now, class, let's look at the second one, which we are told that self-pollinated tall plants planted their seeds and observed selected tall plants obtained pure-bred tall plants. Class, pure-bred, let's look parental phenotype. Parental phenotype. It will be tall, parental genotype. It will be TT multiplied by TT gametes. This will be now fusion. Now, fusion, offsprings. Now, this one will be TT, 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 TT. Now, phenotype of the first fetal generation of the second experiment, he found out that all were tall, but before this one, he selected the pure breed of tall plant. He was having a mixture until he got the pure breeds than himself. Then he got this one. Now from their class, we were told he pollinated again this pure one with this pure bred dwarf with this pure bred tall plants. Now let's look. Now this one we have already discussed. We can rub it, then we can look the way other one. Now we were at tall plants against pure tall plants against now let's look very quickly parental phenotype. Parental phenotype will be tall, dwarf. Now this one will be this. We are taking this one and we are taking this one. The theory we are discussing is a pure bred. We are not non-hybrid. These are pure bred and a pure bred. Pure bred, this one means pure bred. purebred tall, purebred dwarf. This one purebred tall, that is, it will be parental genotype. Parental genotype will be TT. This one will be TT. Now gametes. Now gametes. Gametes will be. Gametes will be. This one will be. This one will be. Now fusion. Fusion of gametes. This one fused like that. 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 Now again, offsprings. Offsprings will be TT, 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 TT. Class, this is fusion, this is fusion, this is fusion, and this is offsprings. Now these are called F1 generation. F1 generation between a purebred tall plant and a purebred dwarf plant. Now you're going to get all the plants to be tall. But again, this one, this tallness is not the homozygous tallness, but rather heterozygous. Where we have the gene for tallness as well the gene for shortness. But the gene for tallness expresses phenotypically in the presence of the gene for shortness because it is a dominant gene over it. Very good. Again, class, he selfed the offsprings between the purebred. Again, he selfed the offsprings from the purebred of tall pea plants and the purebred of dwarf plants. Then what has he obtained? What has he obtained? This one we are done, right? Now let's use this part, then we finish up the question. Now this one will be, now this one we can see all tall but heterozygous. Now parental phenotype. Parental phenotype, it will be, now this one will be heterozygous tall, heterozygous tall. This one will be heterozygous tall as well. Heterozygous tall. This is heterozygous tall plant. This one is heterozygous tall plant as well. Now let's solve them. Parental genotype. This one will be TT, that's the heterozygous for tallness. This one heterozygous for shortness. Now, gametes. Let them segregate. This one, this one, this one, this one. Now crosses. Fusion happens. Fusion. Now, offsprings is now fusion. F2 generation. Now it will be homozygous tallness, heterozygous tallness, heterozygous tallness, recessive, homozygous for dwarfism. Now, let's look at this one very quickly. Now, phenotype. Phenotype will be, this is one tall, very tall, one short or one dwarf. As you see now, this is what he finally obtained. He found very tall and one dwarf. Genotype. Genotype will be 1 TT, 2 TT, 1tt. And that is how this man formulated and was successful. Now, the success of this man sometimes is being attributed to the ways and modalities on which he was working on. This man, first thing he did was, why was Gregor Mendel, the successful scientist, to found the law of heredity, and his law was successful one, while others who have tried failed? Number one, he used favorable materials, e.g. the garden pea plants. The garden pea plants. The garden pea plants class was self-fertilized, and at will he used to for cross-fertilization, as well. Now the pea plant had several observable contrasting traits or other characteristics, and such enabled him to perform multiple characteristics to prove his point. That is why the garden pea plant had several contrasting characteristics. Number one, he selected favorable materials, that is the garden pea plant that was self-fertilized, and he did cross-fertilization or other pollination at will. Again, he focused on a particular trait, while others before him used to do a lot of traits at once, and therefore it wasn't able for them to keep a track record. Another important thing, he kept accurate data of all his experiments. Now, his data he used for analysis. Until then, he formulated a definite hypothesis. Because he kept accurate data, and therefore after analyzing his data, he was able to formulate definite hypothesis class, definite hypothesis. And that is why I want us to stop today. In the next class, we will be discussing about the hybrid and things of the sort. Wish you all the best. With you here is Teacher Muhyiddin Khalif.